Yeah, I'm really happy to tell you about my research entitled Understanding Bee to Hummingbird Pollination Shifts in the Neotropical Spiral Gingers. Kathleen mentioned that I got this fellowship, Faculty for the Future, and I want to acknowledge it first thing because without that support of like 4.5 years, I think I wouldn't be here today. Okay, I want to start talking about Charles Darwin and what is called Darwin Abominable Mystery, which has two parts. The first one is what is the origin of plants with flowers that we call angiosperms. And the second part of this mystery is how come the angiosperms are so diverse and what is the key of their success. And to show you proof that, yeah, angiosperms are a big group of plants on the earth, let's look at this pie chart, simple chart where we can see angiosperms occupy almost 90% of the diversity of plants on the earth. And on this second figure, I want to show you that high diversity of plants with flowers is mainly located in tropical areas in the world. And on the top panel, we have like cooler colors indicate the highest number of plant families. And we can see they're mainly in the American tropics, or what we call the neotropics. And on the bottom panel, we have the number of species, and also the cooler colors are mainly in the neotropics. So the question is, why is there such a great diversity of angiosperms in the neotropics? And that's what I joined this program to try to solve that puzzle. So there's plenty of evidence that show that the interaction of plants with other living organisms, including mutualistic interactions as the one that they have with pollinators, have contributed a lot to plant speciation processes. And there are different mechanisms of how pollinators can mediate speciation processes in plants. And I'm gonna focus my research, or I focus my research in one of those mechanisms. It's called pollination shifts. And that means changes between distant pollination systems happen, and maybe it's mainly through changes in floral traits. So how do we know that this is an important mechanism? So I'm going to tell you about the evidence for pollination shifts. We learn about pollination shifts through phylogenetic studies and fieldwork observations. So from the field observations, we learn what are the pollinators of a specific species, plant species that have specific floral traits. And then with that information, we can infer that those are also the pollinators of plant species with flowers that have similar floral traits. Then we reconstruct the phylogenetic relationships of the plants, and we trace what was the ancestral pollinator of the clade of plants. And then when we see changes, for example, here, when we see a change in the color of the branches, there is when a pollination shift occurs. For example, to tell you more about this figure, this is a phylogeny of the North American clade of columbines. And in this clade, there are species pollinated by bees, by hub moths, and hummingbirds. And when we see a change in color from red to blue, there is, from blue to red, there we can say that pollination shift happened from bee to hummingbirds. So based on that type of evidence, we know pollination shifts are frequent and widespread among angiosperms, and they can happen between different groups of pollinators. As we can see, the arrows here indicate the direction of one pollinator group to another pollinator group. And there is one type of pollination shift that is very common, and it's indicated by the thicker arrows here that goes from bees to bird pollinators. And in the neotropics, where I center my research, this pollination shift shifts go from bees to hummingbirds, since hummingbirds are the most important bird pollinators in the neotropics. So here I'm gonna explain you what are possible scenarios for pollination shifts. Like there's been a lot of studies trying to explain how and why pollination shifts happen. So this is like the background context for my research. So let's focus on this small herb here. It's in this tropical forest. So one of the possible scenarios explains that a plant expands its habitat range to a different place. And then we have like a different population here that is going to be exposed to a different environment, including exposed to different pollinators. So in the original place, it had a pretty rich assemblage of bee pollinators that with which they interact. But in this new place, there are not as many bees, and it's exposed to a different assemblage. So because the flowers then are kind of an unused resource, the new pollinators can start feeding on these flowers, and then they're gonna exert novel selection pressures. And that's how the flowers then are gonna be changing. So this is gonna cause that this population is gonna suffer a decrease in the reproductive success until it gets adapted to the new pollinator. And then we're gonna have a pollination shift happening. So the plant is gonna migrate to a bee pollination adaptive peak to a new one that in this case, we're talking about hummingbird pollination adaptive peak. I also mentioned that pollination shifts happen through floral trait changes. So we have for example, a bee adapted flower that is being used by a bird now, and there are gonna be novel selection pressures. And over time, there are gonna be floral trait changes. And at the end, we're gonna have a different flower. Also, as a result, we're gonna have two different species later. So in this context, there are some gaps of information, which I try to fill a little bit with my research. For example, understanding better what could be promoting ecological factors for pollination shifts, for bee to hummingbird pollination shifts. And then what are floral evolutionary changes involved in a bee to hummingbird pollination shift? And also, there is some information that says that hummingbirds might confer a reproductive advantage to the plants they pollinate, and that are better than the bees. And that's another reason, or that could be another promoting factor for pollination shifts. So those are So what we know, this context is mainly based on temperate study systems, but I'm telling you about the tropics. So should we use this information for tropical systems, or should we study then tropical systems? And I think we have to study tropical systems to achieve a more comprehensive understanding of pollination shifts. These are some of the common temperate system or model temperate system to understand pollination shifts. And there are differences between temperate and tropical systems. And I want to tell you more reasons to why it's important to conduct this research in a tropical system. For example, temperate systems are less diverse environments than tropical ones, and tropical ones are just more dynamic. The distribution of the plants is also different. In temperate systems, there is more patchy distribution of plants or individuals from the same species, but in tropical systems, the individuals from the same species are more sparse. Then we also have different species of pollinators, including bees and hummingbirds. And specifically in tropical systems, because the plants are so dispersed, the pollinators have to do long feeding routes. That's why they are known as trap lining pollinators. And that's why I'm gonna introduce you to this tropical system, the genus Costus, the Neotropical clay, which I use for my research. Let me drink water. So the Neotropical spiral gingers in South America, at least they're known in Spanish as Caña agria. They belong to the genus Costus, the family Costaceae, and there are more than 80 species described within this Neotropical clay. They are big plants, big herbs. They have inflorescence that can be on the tip of the stem or basal inflorescence. They produce one flower per day, but the flower only lasts one day. But their blooming season is kind of long, for like 2 to 3 or 4 months. And so in the Neotropical clade of Costus, we know there are species that are pollinated by euglossine bees, also known as orchid bees, and other species that are pollinated by hummingbirds. And as you can see on these pictures, and these two pollination, the flowers differ a lot between these two pollination systems. Bee pollinator flowers on Costus are bigger, just longer. They have pale colors. They have this big structure that it's called labellum with yellow stripes that function as nectar guides for the bees. And the hummingbird adapted flowers are smaller, are narrower. They have brightly colors, and they don't have the big labellum. Let me play these videos for you, or maybe they don't play. Let me see. There you go. So this is just to show you how the pollinators interact with their flowers in Costus. And another reason why using the Costus system is good for this type of studies is because in the most recent phylogeny published by our former postdoc Oscar Vargas and Kathleen and other collaborators, we know that there are at least 11 independent pollination shifts from bee to hummingbirds that occur in Costus, in the Neotropical Costus clade. Also, I wanna now acknowledge the work of Kathleen for more than 2 decades, like doing research on Costus and positioning the Costus system as a model neotropical system to understand plant evolution. And something else I want to acknowledge is like her decision to bring students from the neotropics so we can study our land or natural resources. And that's like me and my lab mate Pedro Juarez from Costa Rica. So, okay, as I told you, I'm going to try to fill these gaps of information. And so here are my presentation has 3 parts corresponding to different chapters on my dissertation. And on the first one, I'm gonna evaluate if elevation could be a promoting factor for bee to hummingbird pollination shifts. The goal of this part was to analyze floral traits and pollinator assemblage variation along an elevation gradient and whether there is an association between these 2 components. And so why did I choose elevation as the abiotic factor I want to analyze? There are like a couple of reasons for that. First is because there are previous studies that show that plant populations occurring along elevation gradients can show floral variation mediated by the pollinators. And the second reason is because it is known that in higher areas, the abundance of insect pollinators decrease. So there are studies showing the variation of floral traits and pollinator assemblage along elevation in temperate areas, and I wanted to show you one of them. This has been conducted in Norway, and the researchers studying different sites along elevational gradient of up to 1,000 meters. And the species is called Campanula rotundifolia. And here I'm showing you two graphs that plot two different floral traits, the corolla diameter and the flower length. And we can see how these floral traits increase in size with elevation. They also found that the pollinator assemblage is mainly composed of small size bees in lowland areas and bigger size bees in highland areas. Something else that is related to my second point about the abundance of insects in high elevation, they found that the visitation rate of bees to the flowers decreased with elevation, kind of like meaning that there might be less bees in highest areas. So I wanted to replicate a study like this with a tropical system. And I look for descriptions of different Costus species through like herbarium specimens or digital bases like iNaturalist to see if there was any variation of floral traits on any of these species. And I found one that is called Costus guanayensis variety Tharmicus that have different descriptions of floral traits in different elevation areas. So I use Tharmicus to answer these two questions. Let's go see the first one. If floral phenotypes and pollinator assemblages of Tharmicus vary among sites along an elevational gradient. For this, we sampled 4 sites with Tharmicus. The sites were located in Pasco, Peru, around different national protected areas that are indicated here with these different colors. And we sampled 2 sites in low elevational areas, one from 280 to 350 meters, a second one from 300 to 400 meters in elevation. Then we have a third site that we call the middle site between 500 and 700 meters. And our highest site was located between 1,000 and 1,200 meters in elevation. So in all these different sites, we quantify variation in floral traits. For that, we took pictures to analyze the size of the flower and the color on the labellum. And also we took samples of the nectar to analyze the nectar sugar concentration of the flowers. To assess the pollinator assemblages, we set up cameras in the forest with motion detection system to capture the pollinator activity in the flowers. So we proposed 2 different alternatives of how the floral traits and the pollinator assemblages might vary along elevation. The first one we call the local bee adaptation hypothesis. And this is related to the study I showed you at the beginning in Norway, where we proposed that along the elevation, all the sites the flowers are gonna be visited by bees. And the floral traits, we're gonna see an increase in size of the flowers. We're gonna see always conspicuous yellow nectar guides for the bees. And also the nectar sugar concentration may increase with elevation. Our second alternative that we call the pollinator shift hypothesis, we're gonna see a similar changes in the flowers as I explained previously, but in the highest side, we're gonna see something different. We expect to see something different. And there, we expect to see bees in the pollinator assemblage, but also hummingbirds. And because of that, then the flowers might show different floral traits that are more related to hummingbird attraction or a better fit for hummingbird pollination. That is like smaller flowers, the lack of the yellow nectar guides, and a lower nectar sugar concentration. So now I'm going to show you what we found after sampling these 4 sites. Here I plotted traits of flower length on the top panel and flower width on the bottom panel. along elevation. Like I have my 4 sites going from lower elevation to higher elevation. And for the traits related to the floral length, we saw that they increase in length with elevation, but only up to the middle side, and the lowest side have shorter flowers. Regarding the corolla width, the pattern is different, and in this case, we saw an increase on corolla width along the elevation, more similar to the study in temperate areas. So these results, the first ones support or align better with our expectations for pollination, the pollination shift hypothesis. And the second one aligns better with the bee adaptation hypothesis. Then for the flower color analysis, we measure the areas in the labellum that correspond to the nectar guides. That's what we call the nectar guides fraction area. And here we have 3 sites along the elevational gradient. And we found that the nectar guides fraction area was the smallest in the highest site, meaning that these flowers are kind of lacking these nectar guides. So that aligns better with the pollination shift hypothesis. Kind of the lack of nectar guides might be to deter the bee visitation. And final result about floral traits, it's about the nectar sugar concentration, where we found that the highest site had the lowest nectar sugar concentration. And this result also supports the pollination shift hypothesis. So now I'm going to show you our results about the pollinator assemblages. We record different species of orchid bees in the different sites. Some species overlap among sites, but every site also has its own species. And we record hummingbirds visiting flowers in the highest site. This is a table summary of our results. So we have the size from low to high, and we estimated the visitation rates of bees and hummingbirds to the flowers in each site, and we compared these visitation rates. For the bees, we found that only the low side, second low side, had the highest bee visitation rate, and the visitation rate didn't differ among the other sites. And this result might be explained because the low side too, it's surrounded by or is within the national protected areas, like pretty deep inside. And that's like we have a more pristine area where there might be just a lot more orchid bees. That's why the visitation rate is so high compared to the other sites that are like next to rows. They are out, kind of outside or in the border of national protected areas in Peru. So for the hummingbird visitation rate, we also compared this variable, and the high site had the highest hummingbird visitation rate. And in the low side, there was none visit by hummingbirds. We also compare the pollinator assemblages with a non-metric multidimensional scaling. Like it's a way to see how similar or different are the pollinator assemblages among sites. And we found that they are all different among the sites. We also have a result that tell us that in lowland areas, the highest bee visitation rate correspond to bees of a small size, and in higher areas, the highest bee visitation rate correspond to bigger bees. And that is related to my second question, but from this result, I want to say that we found support for the bee adaptation hypothesis because all of the sites have bees as part of the pollinator assemblages, but also the presence of hummingbirds in the highest sites support the pollination shift hypothesis. And for our second question of looking if there is an association between the pollinator assemblages and the floral trait variation. Let's drink more water. So to answer this question, we correlate our measurements of floral traits with measurements we took from the bee body sizes. So for this, I I got help from Doralice Zúñiga Rivas, who captured the bees and who helped me measure the bees' bodies. And we selected one species of bee from each site, which was the species with the highest visitation rate for each of the sites. And Doralice measured the thorax of the bees and also the length of their proboscis, or we can call it tongue. So we correlated these traits. On the top panel, we have the corolla width and the width of the thorax of the bee, and we found a positive correlation between these two traits along the elevational gradient, meaning that lower sites have smaller bees as the main pollinators, and higher sites have larger bees as their main pollinator. Regarding the results for traits related to length, we didn't find a correlation between those traits. So these results support the bee adaptation hypothesis. So there's a mix of results that support one of the other hypotheses. And so with that, I'm gonna conclude this part of my dissertation, saying that we found substantial divergence in floral traits of this neotropical Costus species and its pollinator assemblage across an elevational gradient. We found an association between the floral width and the bee pollinator size, establishing the necessary preconditions for pollinator-driven divergence. And also, Ptarmicus is adapted to the local bee fauna along the study elevational gradient, but we cannot rule out the possibility of the beginning of a bee to hummingbird pollination shift in the highest site. So we're gonna move on to the second part, where I ask what floral trait changes might be involved, or I try to understand what floral trait changes might be involved in a bee to hummingbird pollination shift. So the purpose of this part was to determine which floral traits function as anti-bee and pro-bird traits, meaning that which floral traits might deter bee visitation or preclude bee visitation and good contact with the flower reproductive organs, and which trait might attract more hummingbirds and improve the feed between the hummingbird beak and the flowers. Yeah, this is because floral traits have effects on pollinator attraction and pollinator flower feed. So different floral traits are gonna work for pollinator attraction or pollinator flower feed. So as you can see, species pollinated by bees in different systems have different floral traits from species pollinated by hummingbirds. And there have been studies that modify floral traits in bee flowers, trying to make them look more like hummingbird flowers. And that's how we can test the function of the floral traits as anti-bee or pro-bee traits. I will say it's easier to test for anti-bee traits because we're working with a bee flower instead of testing for pro-bird traits, because it's hard to just with floral surgery trying to make a hummingbird flower. So for this study, we found a species that was very abundant in Costa Rica in this site called La Selva Biological Station from the Organization for Tropical Studies. It's called Costus malortianus. And this was a great species to use because its primary pollinator are the orchid bees, but late in the wet season, malortianus receives hummingbird visits. So having a species that is visited by both group of pollinators was the best for these experiments I'm going to explain you next. So before I go deep into my experiments, I want to show you a little bit about the Costus flower morphology so you can understand better why I did some floral trait modifications. So this is a Costus flower, a bee flower. We have the three petals outside. Then in the middle, we have this modified petal that we call labellum. Is in the bee is the The labellum is white and is very colorful with two yellow stripes that function as nectar guides. And if we do a cross section of the flower, we can see the internal part that has the their reproductive organs. They are attached to a modified stamen that is called the petaloid stamen, is this part. And in the bee flower, it has a very long tip that goes all the way outside of the flower. So here we can see the tip of the petaloid stamen. So I want to show you this video so where we can see how the bee interacts with the flower. It lands on the labellum, on the yellow nectar guides. Then it crawls inside the flower, drinks the nectar, and when it's crawling inside, it deposits pollen on the stigma, and when it's crawling like backwards, it's gonna take pollen from the anthers and hopefully go visit another conspecific flower. So I ran 3 different experiments for this part of my dissertation. In the first one, I wanted to answer this question, like which floral trait modifications decrease the bee pollination efficiency and increase the hummingbird pollination efficiency. And I measured the pollination efficiency counting how many pollen grains the pollinators can transfer between flowers. So for this, I applied 3 modifications. I had a flower treatment where I cut the labellum, so I had a short flower. I had another treatment where I attach a thread around the flower. I call it the constrained flower. And a third treatment where I cut the tip of the petaloid stamen. So I call this treatment the short stamen tip flower. I also apply double modifications at a time, just thinking about flowers as an integral organ. So when they evolve, many traits might change at a time. In the field, what I did was finding big patches of Costus malortianus, and then I applied the floral modification to all these flowers, like the same modification per day. And I waited for the pollinators to come visit the flowers, multiple flowers, so they can transfer pollen between them. And then after a visit, I collected the part of the flower where the pollen gets deposited, that is called the stigma, to count the pollen grains on the stigma. We expected to see that the constrained flower and the short stamen tip flower will lower the pollen transfer by the orchid bees, but all the treatments will increase the pollen transfer by hummingbirds. And for the double trait modifications, I only use these modifications for the hummingbirds just because I didn't have more time in the field. So my results are here. So here we have the results for the hummingbirds and the orchid bees. We have the first box showing our control treatment, which was a natural flower. And then we have the next boxes for each of our treatments. And the y-axis, we are indicating the number of pollen grains we counted on the flowers that the pollinators transfer. So for the hummingbirds, we didn't see an effect of any of our floral trait modification. We didn't see an effect on increasing the number of pollen grains transferred. And in the orchid bees, we did find one treatment that decreased the pollen grains transferred by the orchid bees, and that was the short stamen tip treatment. So this one. So we can say like we found an anti-bee floral trait is reducing the tip of the petaloid stamen. A second experiment we carried out was to answer which floral trait modification attract hummingbirds and deter orchid bees. In this case, we modify the color of the flower with a natural dye, turning it to red. We cut the flower again for a short flower treatment, and we covered the nectar guides with a piece of labellum that came from a different flower. just to make it all the same pattern. So for the design in the field, we had to pot Costus malortianus and locate the potted individuals in different areas in the forest, but we locate them next to a partner species to have our plants within the feeding routes of the pollinators in La Selva, because it was hard to get them, like finding our flowers. So we have different stations, always a control station with the natural flower and stations with our treatment flowers. So there we set up cameras in each station to record the pollinator activity, to see how many of our stations were found by the pollinators and how long did it take for them to find the flowers. And we did this design separately, some days for the orchid bees with this partner species, Calathea, and other days for the hummingbirds with this partner species, it's an Heliconia. And this is how our stations look in real life. There's the partner species, and there are two Costus malortianus flowers next to them, and our cameras in a distance. So for this, we have our expected outcomes compared to natural flowers. We expected that our floral treatments will deter the orchid bees, and only the red flower might function to attract hummingbirds, and no effects from the other treatments. As I was saying, it's harder to, it's easier to test for anti-bee traits than to pro-bee bird traits. So here I'm showing that both the hummingbirds and the orchid bees, they found most of our stations, our control station, and our treatment stations. So they were all good at finding our flowers. And when we analyze or compare the time they took to encounter our stations, for the hummingbirds, we didn't see any effect of our treatments to decrease this time of encounter, especially the red flower didn't decrease this time to encounter. And for the orchid bees, we didn't find any effect either. So none of our treatments increase the time to encounter these flowers in the forest. So this is the final experiment for this part, where we answer which floral trait modifications deter orchid bees, but the experimental design was thought to try to find the mechanisms of deterrence. So we have the short flower treatment, the constrained one, the short stamented flower, and flowers with no nectar guides. And for this experimental setup, we work inside of a tent, and we apply the same floral modification to all our group of plants there. And we waited for the bees to feed on our modified flowers, and we recorded the activity to then analyze how long did it take for the bee to manipulate the flower and enter to the flower to feed on the nectar. So here I have the results compared to the natural flower, and we found that 2 of our treatments increase this handling time by the orchid bees. That was the short stamen tip flower and the no guides flower. They mainly were confused by these changes in the flowers, and that's why they took more time to enter to the flower. So from this part, we found evidence for anti-bee floral traits in the Neotropical Costus, shortening the petalous stamen tip, decrease the pollen transfer to the stigma by the orchid bees, and the same treatment plus removing the nectar guides increase the orchid bees flower access time. And from this part and the first part of my dissertation, I worked with an artist to make a design of how the flowers might change from a bee flower to a hummingbird flower. This is just a hypothesis that I wanted to show you. And so you will see we start with a big flower and it's going to reduce in size over time. Everything's going to be smaller, including the stamen tip, which is going to decrease the pollen transfer by bees, and at some point, maybe increase the pollen transfer by hummingbirds. Then the colors are going to change over the end, the labellum gets reduced, and we have also a narrower flower. So now moving on to the final part, where we carry out a test of the pollination quality between bees and hummingbirds in a number of species beyond Costus species. So the motivation for this part of my research is because studies have highlighted novel consequences of having birds over bees as pollinators in terms of plant reproductive advantage, meaning that birds or hummingbirds might confer a higher pollination quality compared to the bees. And differences in behavior and body size between hummingbirds and bees raise multiple hypotheses about why hummingbirds might be better than the bees. And there are multiple ways to measure pollination quality, but we chose one that was will let us measure it in more species. And that's counting the number of pollen grains that get deposited on the stigma of the flowers. So let's see, if we have a pollinator that visits one flower of Costus malortianus, and then he goes visit one flower from the same species, but from a different individual. So this second flower might get pollen from a conspecific individual. So that is what we call conspecific pollen, gets deposited on the flower stigma. But there's no one species there's no only one species in the forest, but there are multiple species. So in a second scenario, this bee will go to a Costus flower, then to a different species flower, and then to the second Costus flower. So these different species will then get pollen from Costus, which is not its own pollen. So that is what we call heterospecific pollen. So it's pollen coming from a heterospecific individual, not from your same species. And a way to measure pollination quality is to count the number of conspecific and heterospecific pollen on the stigma. That's the pollen grains. So a good pollinator will transfer large conspecific pollen loads and small heterospecific pollen loads to the stigma. And there I'm going to show you a few reasons or a few hypotheses. For example, if we think about the size of the pollinator, hummingbird being bigger than a bee, this hummingbird's large body is going to give more space for different plant species to have a specific placement or their pollen in the hummingbird's body. But a small bee body might not have a lot of room for multiple species, so they all get deposited in the same place. And that is going to cause that different species get a lot of heterospecific pollen. Another trait is the grooming behavior. So there we see a bee that is cleaning itself. So that's a behavior the bees do a lot, even when they are in a feeding room. So they visit a flower, they will clean themselves, flying, they go to another flower. So this behavior of constant cleaning might make the bee carry, in general, a small pollen load. And that would include a small heterospecific load. So that might make the bees better at depositing less heterospecific pollen on the stigma. And hummingbirds don't have that grooming behavior, so they might deposit, in general, bigger pollen loads, including heterospecific pollen loads. Another trait is the floral constancy, which is like a behavior that a pollinator has to just go visit flowers from the same species. So bees have this floral constancy, and that might be beneficial to the plants to get only conspecific pollens. In contrast, hummingbirds do not have a floral constancy, so they will go to multiple species in a feeding route. So to assess the number of conspecific and heterospecific pollen loads on the stigma of flowers pollinated by bees and hummingbirds, we collected the stigmas of different species in multiple sites in Peru and Costa Rica, and we counted the pollen grains and distinguished conspecifics and heterospecifics and the number of different heterospecific pollen. We estimated these variables, a ratio between heterospecific and conspecific pollen, the number of different heterospecific pollen types, and the percentage of samples that we collected that had heterospecific pollen. So we compare these variables between the group of bee pollinator species and hummingbird pollinator species. So here we have our results. The yellow bars or box correspond to bee pollinator species, and the green box correspond to hummingbird pollinator species. We didn't find a difference in the variable, the ratio of HP over CP, neither a difference in the percentage of samples with heterospecific pollen, but we did find a difference in the number of heterospecific pollen types deposited on the stigma, and in this case, the hummingbird transferred more types of heterospecific pollens compared to the bees. So this would mean that both pollinators kind of offering the same pollination quality, except that hummingbirds might deposit more number of heterospecific pollen. A final analysis we carry out with our results from heterospecific and conspecific pollen was to correlate these two numbers, because the relationship between them can tell us about the interactions happening in the plant community that is mediated by the pollinators. So there are three alternatives for the relationship between these two variables. If there is a positive correlation between HP and CP, then we're talking about the pollinators facilitating the pollination in the plants, meaning like if a bee visits multiple species, but each of them still is going to get a higher conspecific pollen grains, and it doesn't matter if it also gets a higher heterospecific pollen load. A second alternative is if there is a negative correlation between these two variables, which is going to be which is an indicator of competition happening in the community. So the plants are competing for their pollinators, so there's going to be a detriment for the plant that gets a lot of heterospecific pollen and little conspecific pollen loads. A third alternative is when there is a balance between these two interactions happening, and there's no benefit or detriment for the plant species, or we can call it commensalisms. So we correlate our two variables, and we found a no correlation when we use the bee species. So there is no benefit or detriment for the bee pollinator species. In contrast, we found a positive correlation when we use the hummingbird species, meaning that the hummingbirds are facilitating the interactions in the plant community. Yeah, so to close this part, we conclude that bees and hummingbirds offer a similar pollination quality in terms of heterospecific pollen load sites and the occurrence of heterospecific pollen. The pollinator groups only differ in that hummingbirds transfer a higher diversity of heterospecific pollen types to the stigma than bees. We show a neutral outcome for the plant-to-plant interaction in the bee pollinator community, and a positive outcome or facilitation in the hummingbird pollinated community. Okay, so with that, I try to cover all these gaps. And let's go to the summary. Sorry. I'm going to skip my joke. So as a summary for my dissertation, our findings contribute to understanding B2 hummingbird pollination shifts beyond temperate systems. We provide evidence or insights on the effect of elevation on pollinator assemblages and floral divergence. We elucidated why floral trait changes might be involved in the transformation from a bee to a hummingbird adapted flower. We show that hummingbird pollinators might not confer a higher adaptive peak compared to bee pollinators. So overall, I think we contributed to understand evolutionary processes that promote species shifting mutualistic partners like pollinators, and thus processes that increase the diversity of life. And that's the end